Mitochondria or singular mitochondrion. They are specialized organelles which can be easily seen under the microscope, and these the number of mitochondria, it varies in different cells according to the functions of the cell. They are cylindrical, they can be sausage shaped, their diameter is generally 0.2 to 1 micrometers, average 0.5 micrometers, and their length is 1 to 4 micrometers in length. So mitochondria, they are double membrane structures, their outer membrane and the inner membrane. When we see the inner membrane, we find that it has folds. Now these folds form two compartments, that is the outer compartment and the inner compartment. The inner compartment is called as the matrix, and the outer compartment forms a continuous limiting boundary of the organelle. So the outer membrane forms the continuous membrane, and the inner membrane is folded, and thus they form matrix and the cristae. So these folds, they are called as the cristae, which are towards the matrix, and these cristae actually increases the surface area. So the two membranes, they have their own enzymes which are associated with the mitochondrial functions. So they are the sites of aerobic respiration, and they produce cellular energy in the form of ATP. So they are also called as the powerhouse of the cells, and the matrix also possess single circular DNA molecule. So this here is single circular DNA that is also present here, you can see here, and a few RNA molecules also. Ribosomes are of the 70S type. So their ribosomes are of 70S type, whereas in a prokaryotic cell we know that they are of the 50S type. So their components, for the protein synthesis, these components are required, and they also divide by fission. Plastids. Plastids, they are found in the plant cells and in the euglenoids, and they are easily observed under the microscope as they are large. They can bear some specific pigments, and they impart specific colors to the plant. So based on the type of the pigments, plastids can be classified into chloroplast, chromoplast and leucoplast. These are the main three types. So the chloroplasts, they contain chlorophyll, and when we talk, there are various other types which are for the storage of food as well as containing other pigments, providing pigments to the different colors in the plant. So when we talk of chloroplasts, they contain chlorophyll and carotenoid pigments which are responsible for the trapping light energy, which is essential for photosynthesis. In the chloroplast, fat molecule that is called as carotenoid, which is a pigment like carotene, xanthophylls and others, they are present, and this gives part of the plant a yellow, orange or red color. The leucoplasts are colorless plastids. They have various shapes, and they store nutrients also. We can see the previous slide. Here we can see these are the leucoplasts, and amyloplasts, they store fats or starch, where like in potato, and elaioplasts, they store oil and fats, whereas aleuroplasts, there are other types which are aleuroplasts also, and they store proteins. So majority of the chloroplasts of the green plants, they are found in the mesophyll cells of the leaves, and they are lens shaped, oval, spherical, ribbon like also in some, and their length also varies. And like mitochondria, chloroplasts, they also have double membrane. They are membrane bound organelles. So their inner chloroplast membrane is less permeable, and the space in the inner membrane of the chloroplast is called as the stroma. And here we see a number of organized flattened membrane sacs which are called as thylakoids. They are also present in the stroma. Thylakoids, they are the stalks like coils of the coin, and these are called as the grana, and singular is granum. Or they are integrated thylakoids also. They are flat membranous tubules which are called as the stroma. This is the stroma. stroma lamellae also, which are connecting the thylakoids of the different grana. So the membrane of the thylakoids, they have a space which is called as the lumen. And the stroma of the chloroplast, it contains enzymes which are required for the synthesis of carbohydrates and proteins. It also contains very small double membrane circular DNA also. DNA and ribosomes also. So the chlorophyll pigments are present in the thylakoids, and the ribosomes of the chloroplast are smaller, that is 70S type, and their cytoplasmic ribosomes are of 80S type. Ribosomes we have talked about earlier also. Ribosomes, they are composed of RNA and proteins, and they are surrounded by a membrane, and the eukaryotic ribosomes are 80S, whereas the prokaryotic ribosomes are of 70S type. So here S stands for the Svedberg unit, which is for sedimentation coefficient, and it is indirectly is a measure of the density and size. So both 70S and 80S ribosomes are composed of two subunits. Cytoskeletons. Now in cytoskeletons we find the filamentous proteinaceous structures. They are present in the cytoplasm. These are called as cytoskeleton. The cytoskeleton in a cell is involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. Cilia and flagella. Cilia, which is singular, is called as the cilium, and flagella, singular, is called as flagellum. These are hair-like outgrowths of the cell membrane. Cilia, they are small structures which works like oars, and they cause the movement of the cell and the surrounding fluid, whereas flagella, they are longer and they are responsible for the cell movement. The prokaryotic bacteria possess flagella, but they are structurally different from that of the eukaryotic flagella. So the electron microscope study of the cilium or flagellum shows that they are covered with the plasma membrane. They are covered with the plasma membrane, and they are also, their core is called as exoneme. So their core is called as exoneme, and they possess a number of microtubules which are running parallel into the long axis. The exoneme usually have 9 pairs of doublets. They have 9 pairs of doublets, and these pair of centrally located microtubules. They have centrally located these microtubules also, and this arrangement of the exoneme microtubule is referred to as 9+2 arrangement, and the central tubules are connected by bridges, which is also called as the sheath, central sheath, and this is connected to the tubules of each peripheral doublets by a radical spoke. Okay, these are radial spokes, and the peripheral tubules, they are also interconnected by linkers. They are also interconnected by these linkers, you can see here. And both the cells and the cilium and the flagellum, they emerge from the centriole like structures which are called as basal bodies. So both cilia and flagella, they originate from basal bodies. Centrosome and centrioles. Centrosome is an organelle which is usually containing 2 cylindrical structures which are called as centrioles. They are surrounded by amorphous pericentriolar material, and both the centrioles in a centrosome is perpendicular to each other, and each has an organization of a cartwheel, and they are made up of 9 evenly squared spherical fibrils of the protein which is called as tubulin, and each of the peripheral fibril is a triplet. So here you can see the structure of the centriole. These are the centrioles, which is a mother centriole and the daughter centriole with the interconnected fibers with the proximal and the distal ends with the microtubule triplets. The adjacent triplets, they are also linked, and the central part of the proximal region of the centriole is also proteinaceous, which is called as the hub. Like here, this is called as the hub. and these are the triplets of the centriole. This is the basal body, and when you see the inside, you see that they are arranged in the form of a triplets, and they are connected to the peripheral triplets by the radial spokes also, which are made of proteins. And the centrioles from the basal body of the cilia or flagella, they are spindle fibers, and they give rise to the spindle apparatus during the cell division in the animal cells. Nucleus. Nucleus is an organelle which was first discovered by Robert Brown, and the material of the nucleus, which is stained by the basic dyes, is known as chromatin. So chromatin is the inner thread-like material. The interphase stage of the nucleus, when it is dividing, it is highly extended, and it contains the nucleoprotein fibrils, which are called as chromatin. And these chromatin they change into chromosomes at the time of cell division. So apart from chromatin, nuclear matrix and the other spherical bodies, which are called as nucleoli, or singular is nucleolus, they are also present. They also have a nuclear envelope, which consists of parallel membranes and space between them, which is called as the perinuclear space. It forms a barrier between the materials present inside the nucleus and that of the cytoplasm. So the outer membrane usually contains endoplasmic reticulum, and it also bears ribosomes in it. So the number of places the nuclear envelope is interrupted by many pores, which are formed for the fusion of the two membranes, and these nuclear pores are the passage through which the movement of the RNA and protein molecules takes place in both directions between the nucleus and the cytoplasm. There is only one nucleus per cell. Variations in the number of nuclei, they are also observed, and in eukaryotes, in erythrocytes of many mammals, and some of the tube cells of the vascular plants. So there we find the variations in the nucleus, they are there. So nuclear matrix or the nucleoplasm, it contains nucleolus and the chromatin, and the nucleoli, they are spherical structures that are present in the nucleoplasm, and the content of the nucleolus is continuous with the rest of the nucleoplasm, and it is the site of the active ribosomal or RNA synthesis, and there are more numerous nuclei which are present in this site, which helps in protein synthesis. Now nucleus, when undergoes division, or the cell when undergoes division, chromatin changes into chromosomes. Chromatin contains DNA. Here you can find these thread-like structures. These are the DNA which is present in the chromosomes. Now chromatin contains DNA and some basic proteins which are called as histones, and some non-histone proteins also along with the RNA. So a single human cell has approximately 2 meters long thread of DNA, which is distributed along its 46 or 23 pairs of chromosomes. So the details of the DNA we will be studying in class 12. So every chromosome essentially has a pair of centromere, on the sides of which the disc-shaped kinetochores are there. You can see here kinetochores. They are present at the sides of the centromere. So based on the position of the centromere, the chromosomes can be divided into 4 types. Now these are the types of chromosomes. First type is metacentric, which has the middle centromere, like here centromere is in the middle. These are called as metacentric chromosomes, which have 2 equal arms. Submetacentric chromosome has centromere slightly away from the center, from the middle of the chromosome, resulting into one shorter arm and one longer arm. And in the case of acrocentric chromosome, the centromere is situated close to its end, forming one extremely short and one very long arm. Whereas in the telocentric chromosome, we find that one extremely short and one extremely long arm, whereas in the telocentric we have a terminal, it is situated at a terminal centromere. So sometimes a few chromosomes have non-staining secondary constrictions, which is at a constant location.